Hi, this is my third hat. I was lucky enough to go to Cirque du Soleil one time and I got this cool hat there. Let's talk about chapter 14. Chapter 14 is the most complicated chapter of the new material for the end of the semester. I think this worksheet has pretty self-explanatory answers, so I'm just going to go over a couple of things on it. The first one that I want to talk about is question 1. The link between NADPH and the pentose phosphate pathway and the damage to cells. So what that's referring to is this part of the pentose phosphate pathway that's at the top. So this part here. And what happens in this first set of steps is NADPH is made. That is very similar to NADH. So it's a molecule that basically harbors electrons and can be used as a reducer. So the critical thing in terms of preventing oxidative damage to cells is to have plenty of this NADPH available, because it is used by glutathione reductase. So here's the first step of the pentose phosphate pathway. It's done by glucose 6-phosphate dehydrogenase, and it creates a molecule, actually two molecules when it goes through the whole set of reactions of NADPH. So this molecule then is able to reduce, and it's used by glutathione reductase to reduce a disulfide bond here to the reduced form where you have the two free SH groups. So here it's oxidized. If you use glutathione reductase, it reduces it to GSH and GSH, two molecules of that from one disulfide bond. And the glutathione reductase has to have NADPH in order to do that reaction. So why is that important? That's shown in the bottom part of this diagram. So the important thing is glutathione peroxidase, which is another enzyme, uses the reduced glutathione to carry out this reaction. So hydrogen peroxide and other reactive oxygen species like the superoxide radical, which is shown here, oxygen itself, hydroxide radical, which is not shown, all of those things can lead to a high production of hydrogen peroxide, which is an oxidizer in your body, and it can cause damage to your cells, especially to nucleotides, so damage to RNA and DNA, and some proteins and fatty acids. And if you have too much of that damage, your cells start to die. So glutathione peroxidase will convert hydrogen peroxide to water and detoxify your cells from buildup of these reactive oxygen species. So in order for glutathione peroxidase to keep working, it has to have a source of reduced glutathione, and that's where glutathione reductase comes in. It does not directly impact this part of the detoxification process, but it just keeps regenerating the coenzyme that glutathione peroxidase needs. So both of these have to work in order for your cells to stay detoxified and not be damaged by oxygen reactive species. And where do we get the NADPH? That's where the pentose phosphate pathway comes in. So all three of these enzymes have to work together to keep your cells detoxified and protected from oxygen reactive species, which are normally occurring and then in some cases can be excess and cause extra damage. The other part of this pathway is shown here, and what happens is ribulose 5-phosphate is converted through the action of transketolase and transaldolase to glyceraldehyde 3-phosphate and fructose 6-phosphate. So there's a lot of stuff going on in here that is shown in the slides and in your textbook that you don't really have to worry about. Just know that these two enzymes shuffle a lot of carbons from ribulose 5-phosphate, and you end up with fructose 6-phosphate and glyceraldehyde 3-phosphate. Everything in a box is important. So the fructose 6-phosphate then can be isomerized to glucose 6-phosphate, and what happens to that depends on what's going on in the cell. So if NADPH is low or your cells are under oxidative stress, the glucose 6-phosphate will go back up here and you'll make more NADPH, and that will be used to detoxify cells. If that's not the problem and you have a large source of carbohydrates, glucose 6-phosphate can run through glycolysis and make ATP, or if your energy charge of the cell is low, you could make ATP from glycolysis with this. This is the second intermediate in glycolysis after glucose. The other thing I didn't mention here is ribose 5-phosphate will be made if nucleotides are low in your cell and you have to synthesize them to make DNA and RNA. So whatever happens in the cell is largely dependent on the conditions. If nucleotides are low, ribose 5-phosphate will go this way. If your cells are under oxidative attack, the ribose 5-phosphate will be shuffled and converted back to glucose 6-phosphate for another round of NADPH production. And if the energy charge of the cell is low, you can run the glucose 6-phosphate through glycolysis. The next question I want to talk about is question 4. So here, the importance of this is to understand what affects PFK1. So that's the enzyme in glycolysis, that makes fructose 6-phosphate into fructose 1,6-bisphosphate. So it's that enzyme there. So in order for that enzyme to be active, you have to have a source of fructose 2,6-bisphosphate, and if that's present, glycolysis runs. So PFK2 is the enzyme that makes fructose 2,6-bisphosphate. So if this enzyme's active, you'll have a lot of this compound. PFK will be active and glycolysis will run. However, this question asks you what happens if you get rid of all the PFK2. In that case, you are not making fructose 2,6-bisphosphate. PFK will not be active, and its opposing enzyme, fructose bisphosphatase 1, will be active. And the opposing enzyme is across from it in this diagram. So that is here. The blue and running in this direction is gluconeogenesis. The red running in that direction is glycolysis. So any regulatory enzyme in glycolysis, if it is not active, its opposing enzyme will be active. So in this case, we're asking about this one. If it's not active because that effector, fructose 2,6-bisphosphate, is missing, this enzyme will be active and you will run gluconeogenesis. The last question is asking about epinephrine, and it was just kind of a thought question to see what would happen. So you can look at the last slide in chapter 14. There's a nice diagram there of what epinephrine affects and in different tissue types. So that's kind of summarized here. And then the last question was insulin and glucagon. So those are not directly impacted by dumping of epinephrine into your body, but after you fight or flee, you would have to recover from that. And in those cases, those hormones could be important. So that was what I was getting at the last part of that question. So just kind of think about, depending on what's going on in your body, which hormones would be dumped in and what would their impacts be on carbohydrate metabolism. And the 4 things you should be familiar with are glycolysis, breakdown of glucose, gluconeogenesis, the synthesis of glucose, the synthesis of glycogen. When you have a lot of glucose, you make it into the glycogen polymer. And then when you need glucose, you break down that same polymer to release glucose.